raise your hand if you had the flu this season. Glad to see you all recovered. So we know that humans get infected by viruses, but did you know that bacteria also get infected by virus? These are called bacteriophage, and it literally translates to bacteria eat. So these bacteriophages, they latch onto bacteria, hijack its resources, make multiple copies, and then kill off the bacteria and look for its next prey. So bacteria keeps developing tools to fight against these viruses, and some of these are the same tools used by us to fight against our viruses. So STING pathway is one of the tools that's used by both of them. It has the STING protein at the center of it, and it's used by both bacteria and humans to fight against their respective viruses. However, because we are separated by millions of years of evolution, our stings are a bit different. While the bacterial sting is the basic version, the human sting is more advanced with more cooler features. So I work on the bacterial sting, where it all started. So how does the sting protect bacteria against bacteriophage? So here's a graph which shows how healthy bacteria grows. It keeps multiplying, and so the growth goes up. If the bacteria is infected, you'll see in blue that the growth goes down. So at point A is when bacteria is getting actively infected, the bacteriophage multiplies, and then it goes to and spreads to the next bacteria. So ultimately, all bacteria get infected, and by the time you reach point B, all bacteria die. However, if bacteria has sting, you see at point A, the bacteria that are infected, they make the crucial decision to sacrifice themselves before the bacteriophage gets a chance to spread. So although they die, which is why at point A, you can see that there's a dip in the growth, the rest of the bacteria survive, and then they can grow happily in a bacteriophage-free environment. So sting kills infected bacteria. How does it do so? I use this magenta dye, which cannot get inside bacteria. However, if the bacteria has holes or pores in it, it manages to get inside and color the bacteria magenta. So when we looked at the infected bacteria with sting, we saw that most of them were magenta in color. So this shows that sting seems to be punching holes or pores in the bacteria, here depicted by the pickaxe, and that's how it's killing the infected bacteria. So the human sting is a bit more efficient. So when it encounters a virus, it usually calls for reinforcements, and they get rid of the virus for it. So when you have both the stings next to each other, you'll see that they're very different. However, you'll notice one thing in common. Both of them still hold the pickaxe. So imagine how important this pore formation function is that has been retained over millions of years of evolution, and something as simple as bacteria and as complex as humans, we both use it for our immunity. And because we had been so focused on all the networking that human sting does, that we overlook the fact that it can also be doing the dirty work itself in antiviral immunity. So I study how pore formation works in bacterial sting, which helps me understand what its role is in human sting. So you can appreciate how important bacterial models are for studying this pore formation function, because if you notice, that's the only function that the bacterial sting has. So we can look at it in isolation from all the other complexities and all the other functions that the human sting has. So once you understand what the role of this function is, you can then develop therapeutics that target this specific function, and then you can modulate it to improve antiviral immunity in humans. So the next time you recover from flu, I want you to take a moment to think about all your bacterial ancestors that gave you all these tools to fight it. Thank you.